We just finished talking about replication, making a copy of your DNA so that you can usually split your cell, make a copy. Another process that we use with our DNA is reading it. Remember, it's an instruction manual. If I want a recipe for, making chocolate chip cookies, I can look in my instruction manual and look up chocolate chip cookies and the recipe is there. The other thing that we have to be able to do with our genetic material is read it and use the information that is inside of it. That is a process called transcription and translation. DNA holds all of the information from the cell, for the cell, but it must be transcribed and translated to produce anything within the cell. If we're talking specifically about a eukaryotic cell, remember that our genetics, our DNA, our chromosomes are inside the nucleus. This is like having a book in a library that is a reference material that you can't check out. How do you get the information from that book to your house? You can take notes, write it down, take it home with you, but the book stays where it's at. I hope that makes sense. Transcription, the DNA is transcribed into a complementary strand of something called messenger RNA or mRNA, which is an intermediate between DNA and the protein synthesis machinery. The mRNA holds the blueprint for making the proteins. I want the recipe for chocolate chip cookies out of my grandmother's cookbook, but my grandmother has that cookbook and she's not going to let me take it home. So what do? I go to her house and I write out the recipe. Then I can take the recipe with me home. The book stays where it's at and I've got a copy. Is it an exact copy? Probably not. And let me explain why. Because my handwriting and my grandmother's handwriting are completely different. That's one. Two, my grandmother may use different language or abbreviations that I'm unfamiliar with. For example, in her recipes, a tablespoon isn't written out as TBSP, tablespoon. I'll write it down. So in my grandmother's cookbook, it might have this or it might have this, which for me personally doesn't mean anything. So when I write it down, I might write, which to me means tablespoon, while this means tablespoon to her, whereas this to her means teaspoon. So I might write it like that, meaning I write it slightly different. mRNA, remember, RNA has uracil. It does not have thymine. When I write the complement to the sequence that I am transcribing, it's going to be a little different. It's still the same recipe. It's just my own handwriting. It's my own abbreviations. When the DNA is transcribed into messenger RNA, it's still the same message. It's just that instead of having thymine, because it's DNA, I will have uracil in thymine's place because it's RNA. That intermediate is what the ribosome can read and make into something else. So that's why we say it's an intermediate. DNA cannot come out of the nucleus of a eukaryotic cell. It's trapped in there. Having this way to make take notes on that section and moving it to where my machinery that can actually make something out of it is beneficial, because it guarantees that my chromosomes stay safe inside the nucleus. They're not going to be mutated, they're not going to be cut, they're not going to tangle, they're literally safe. Just like reference books in a library, usually those are very rare or they're very expensive. So we don't want them outside of the library. That's why we don't take them anywhere. We keep them where they are. That is basically the same concept of why DNA stays in the nucleus and we take notes and then those notes leave. The mRNA that we just had the notes for holds a blueprint for making a protein. So the recipe that I just copied from my grandmother's cookbook is an instruction manual on how to make chocolate chip cookies. This is the instruction manual. That piece of mRNA is the instruction manual for how to make a protein. All right, so that's transcription, going from DNA to RNA. Transcription, going from DNA to messenger RNA. Translation is different because translation, we are going to take our instructions, our mRNA instructions, our recipe, and we're going to produce the protein that's in that mRNA strand. I'm going to take the recipe home and make the cookies. So in translation, I'm going from mRNA instructions to a protein. So we have two processes with transcription and translation. Transcription, I'm taking notes off the DNA and moving it to machinery that take that mRNA and make it into a protein. I go to my grandmother's house, take notes on making her chocolate chip cookies, take those notes, take the notes home and make the cookies. mRNA, cookies, mRNA, protein. Let's begin with transcription. DNA is transcribed to messenger RNA. Remember, mRNA is messenger RNA, using the enzyme RNA polymerase. Remember I said DNA polymerase. Polymerase means to make. So RNA polymerase is going to make RNA. The strand of DNA is transcribed. The strand of DNA that is transcribed is known as the template strand. A few key points about transcription. mRNA is single-stranded. That double-sided ladder that we see with DNA isn't how this works. It's literally a single or half a ladder strand. As I said previously, RNA contains uracil instead of thymine. When I was talking about giving you a sequence, this is what I meant. Let's see, DNA, we said Alice, Texas, go Coyotes. Well, RNA instead of the thymine, now we have uracil in its place. Guanine and cytosine are still together though. You will always know which sequence or what you are looking at if I give you a sequence because of the presence of uracil or because of the presence of thymine. Thymine is always DNA, uracil is always RNA. As you can see, we have the steps. Let's go through them individually. Initiation. Transcription is initiated when RNA polymerase recognizes a segment of DNA called a promoter region. This region consists of two sequences of DNA just prior to the beginning of the gene to be transcribed. These promoter sequences provide the signal for RNA polymerase to bind to the DNA. Then there's a special codon called an initiation codon. Let's talk about what a codon is. A codon is a 3-letter sequence. Each 3-letter sequence encodes a specific amino acid or a specific action. There's a start 3-letter sequence or start codon. There are 3 separate 3-letter sequences that mean to stop. So there are 3 stop codons. And then there are the other sequences that each encode a specific amino acid. Remember, we talked about proteins being made up of amino acids. The Death Star is built with individual Legos. Proteins are built with individual amino acids. So the codon that we're talking about is the initiation codon. It's a code that says, RNA polymerase, you start here. This is where RNA polymerase starts its transcription. As the DNA helix unwinds, the polymerase first pulls an early part of the DNA into itself, a process called DNA scrunching. So literally, you can see this here. It's almost like having a spring in a pinball machine. You got the ball in the pinball machine, you pull back the handle and the spring kind of pulls back and it gives it the momentum to hit the ball, right? So that the ball goes into the pinball machine. This scrunching is like the spring in the pinball machine. It gives it the energy, okay, to kind of push, to push down the strand of DNA, to advance down the DNA strand, to continue synthesizing the RNA complement to the template. The nucleotide sequence of the promoters differs only slightly from gene to gene, with all of the promoters being rich in adenine and thymine. This goes back to that 2 bonds versus 3 bonds thing. 2 bonds are easier to break apart than 3. Having an AT-rich region means that it's easier to pull apart so that I can get in here and read my strand to make my mRNA notes or copy. Remember, mRNA, messenger RNA. It's carrying a message, obviously. Only one strand of DNA is the template and is copied by RNA polymerase. Remember that we have reading in the right direction and then reading backwards. So one of the 2 strands is the one that is in the right direction for RNA polymerase to read to make my notes, my messenger RNA from. The second step is a step called elongation. And it's exactly what it sounds like. As my RNA polymerase starts going forward, it starts adding RNA to the template that it's reading. Now, this is another cool thing about this. Unlike DNA replication, where I unzip the whole thing and make a copy of the whole thing, I'm only reading a section of it. If I've got a whole cookbook, I don't write down the whole cookbook for the cookie recipe. I only go to the page that I need and copy down that one page, correct? So here, we're basically doing the same thing. We're unwinding the section that we need and making our mRNA copy. Once I'm done with that, do I leave the cookbook out at my grandmother's house? No, you don't, because what happens? Chancla, that's what happens. So what do? When you're done, you close it, you put it back where you found it, yes? Here, it's the same thing. It unzips, but as it reads, this piece that I've already read starts to coil back into itself. Literally, I'm unzipping and making as I go, and The minute that I'm done, it coils back in or rewinds into its helix or twisted ladder pattern. This reading happens in the 5 to 3' direction. Just like our continuous strand in DNA replication, this is the correct way to read this strand to make the right mRNA transcription. Also, remember that uracil is used in place of thymine. What that means is, so if I have a sequence of DNA, TAC, CAT, GGC, CCA, for RNA, it's A, U, C, G. Across from T is still an A for RNA. All I have that can go across from T in my pool of options is the adenine. So adenine still goes across from thymine. But if in the DNA, let me do this. If in the DNA, I'm going to switch to blue for the RNA. In the DNA, I have an A. What goes across from it is a uracil, because this is messenger RNA. C and G are always the same, regardless of which option I'm choosing. In this case, I'm making messenger RNA. C still has G across from it. A has uracil. T has adenine. Again, this is literally the only option I have in my pool of nucleotides that would fit across from thymine. Guanine has cytosine. Cytosine has guanine. Adenine has uracil. So the complementary pairing is still happening. The only difference is, if with a DNA sequence, I would normally put thymine. Instead, I'm going to replace it with uracil because I'm not making DNA, I'm making RNA. The third step in transcription would be termination. If I go to my grandmother's house to look at a recipe book to get chocolate chip cookie recipes, I don't read all the recipes. There's no reason to. Literally, once I get to the end of the chocolate chip cookie recipe, I can close the book once I've taken my notes and I'm done. Here, it's the same thing. At termination, the RNA polymerase recognizes a stop codon. Again, each 3-letter sequence is specific. It basically has a specific meaning. The polymerase, the RNA polymerase, recognizes a code that signals the separation and release of my mRNA strand, that's what this is, from my transcribing, from copying. Once I get to the end of the recipe, there's no reason to keep going. Once I do that, once I am finished, once I've stopped, ouch, I may have something that's fairly short, maybe 100 bases long. I might have something that's a little bigger, 1,200. But really and truly, depending on what we're building, some proteins have mRNA sequences that are several thousand base pairs long. Proteins can be very complicated 3-dimensional structures that have moving parts. And if you've got something that looks like that, it's not going to be 100 bases. It's going to be more complex than that and need a lot more parts. I can build a skateboard probably with, I'll say 20 parts, Never built a skateboard, but just go with me on it. And it's a way for me to transport myself from point A to point B. However, if I was going to build something more complex like a car, let's say 20 pieces isn't going to be enough to build that. It's the same here. The more complex the protein you're building, the longer this instruction manual sequence has to be.